start working on. So it's a new project, and I don't have a lot of data yet, but hopefully it will be interesting for everyone to share it. So as you can all see from the title of my slide, what I've been working on is the molecular evolution of snake photoreceptor development genes. So people might ask, why am I interested in studying snake visual system? Well, the answer is the fossorial lifestyle of ancestral snake has led to degeneration in the visual system of snake. However, nowadays snake managed to adapt to the diverse ecosystem across the world, and many of these ecosystems have different visual ecological conditions. What this suggests is there is important visual adaptation occurred when snake recolonizes terrestrial habitats from their fossorial ancestral stage. And among these important visual adaptations, one particularly interesting adaptation is the evolution of photoreceptor compositions. So shown here is the photoreceptor composition in different snake groups. And at the left side, it is the Hinophidian snake retina. It's a retina composition for Hinophidian snake. And you can see, so it's for python snakes such as python and bows. And it has cone photoreceptor as shown here and rod photoreceptor. So it seems like it has a duplex retina similar to most vertebrates, including human. But when you take a closer look, you can notice that they actually lack a few cone photoreceptor. So it is a reflection. So it's actually a reflection of a partially degenerated photoreceptor composition inherited from their fossorial ancestors. But for Sanophidian snake, for the more advanced Sanophidian snakes, they have a wide variety of different retina compositions, including the Sanophidian type duplex retina, which have more cone photoreceptor types, and also intermediate retina, which is the most interesting one. So these photoreceptors, they are neither cone nor rod, but they resemble, they are kind of in between. And they are also all cone retina and all rod retina for nocturnal and diurnal species. So this very diverse photoreceptor composition, it is not expected, and it is very uncommon among vertebrates. So it is believed that this very high diversity, this diversity of photoreceptor composition in snake is caused by something termed photoreceptor transmutation, which is the evolutionary process by which one photoreceptor type being converted to another. For example, rod converted to cone, or cone converted to rod. And to figure out what kind of gene evolution is behind the evolution of this photoreceptor transmutation, well, currently studies have been done on the snake phototransduction genes, and what they found is for snake phototransduction gene, there are accelerated evolution, especially among Sanophidian clade, which, as I shown in the previous slide, Sanophidian snakes are known for their diverse retinal compositions. So this suggests there is likely involvement of phototransduction gene in the photoreceptor transmutation. But besides phototransduction genes, photoreceptor development genes are also key to the photoreceptor cell type, and also it is expected to be important to the photoreceptor transmutation, because transmutation in the end is about the cell types, and those developmental genes determine the cell types. But unfortunately, there isn't any study that have been currently focusing on the photoreceptor developmental genes. So for my project, I have picked 6 photoreceptor developmental genes from the genetic network regulating photoreceptor development, and so these are the 6 genes and their functions, and I aim to figure out whether there is accelerated evolution in snake photoreceptor development genes, and if yes, is the evolution of the developmental genes associated with photoreceptor transmutation events. In terms of the approach I will use to address this question, I will use the analysis of selection pressure, which basically measures the ratio of non-synonymous to synonymous substitution, and also known as DNDS. And when DNDS is greater than 1, it indicates a positive selection or adaptive evolution, while when the DNDS is far lower than 1, it indicates purifying selection or conservation of functions. So with this with this measurement, I can assess the evolutionary rate and if there's any adaptation in the photoreceptor development genes of snakes. And currently, there are a lot of tools for, and there are a lot of tools or programs for such for the analysis of selection pressure. And for my project, I specifically choose to use phylogenetic analysis by maximum likelihood, and more specifically 2 models implemented in PAMO, which are called clay model D and clay model C. So those models allows me to estimate DNDS ratio for estimated a separate DNDS ratio for the target species and the rest of the species in the phylogeny. For example, let's say in this case, the red species represent the target group, and the rest of the black species represent the non-target group. And clay model D will allow me to estimate a DNDS ratio for the target group and another DNDS ratio for the rest of the species, so I could compare them. And here are the species and sequence sampling. So around 400 sequences are collected for 6 genes from online databases or pulled out from available genome and transcriptomic dataset. And the sequence dataset cover around 100 species from all major Phyllosida clades, as shown in these trees. And in terms of the result, what I've found is there is accelerated evolution in the photoreceptor development genes. And before I go into detail about this specific data, I'd like to first explain how to read these figures. So I will use OTX2 as an example. So here it shows the name of the gene, and here it shows the target group used for each analyze. For example, for this first data point, it basically means for serpentes, or for all snakes, their DNDS ratio is estimated to be so they are the target group, so they are represented by the foreground, the black dot, and their DNDS ratio is estimated to be around 0.08, and the rest of the Sauropsida species when estimated together, they got a DNDS ratio of more than 0.2. And from this figure, what we can observe is there is for NRL and phase 1, they show little to no shift in selective pressure, while OTX2 analyze show stronger purifying selection in snakes, which is pretty unexpected. But for CRX, NR2E3 and NR1D1, they show obvious elevation in DNDS ratio, suggesting potential adaptive evolution. And also one important takeaway from this figure is this accelerated evolution pattern identified in the photoreceptor development genes matches what have been found in the phototransduction genes of snakes. So this together suggests similar to phototransduction genes, photoreceptor development genes of snake might also involved in the evolution of photoreceptor transmutation. To further confirm this hypothesis, I have also done analysis on specific branches or clade within the phylogeny. So here I've shown a phylogeny of snake used in the study, and so here is the phylogeny, and also it is color coded for the photoreceptor compositions. And as you can see, there is clearly several transmutation events occur within this phylogeny. For example, at here, as this branch represents the Cynophidian clade, so it is a basal branch for Cynophidian clade. And here there's a transmutation event occurred that transmuted the ancestral form of Cynophidian duplex retina into the more advanced form of Cynophidian duplex retina. And similarly, at here, another transmutation event, and eventually at here and at here. So there's multiple transmutation event occur within the phylogeny. And since given this multiple transmutation event occurred within the phylogeny, what I can do is I can test I can analyze the DNDS ratio for these branches and clades that have transmutation event is occurred. And what I have found is for the basal Cynophidian branch, there is accelerated evolutionary rate as shown at here. So there's positive selection and also an elevated DNDS ratio. And similarly for the clubroidae branch and clubridae branch, which are, so this one is the clubroidae branch and this one is the clubridae branch. Their names are pretty similar, but there are two branches. And also for the clubridae clade, as shown, so this entire one represents clubridae clade. So for these two branches and also this entire clade, there is positive selection, actually very lot of positive selection, and some of them has got very high DNDS ratio are identified. And finally, for the Lamprey-Fettini clade, as shown at here, we have also found positive selection in the 3 photoreceptor genes, photoreceptor development genes tested. So we have only tested 3 developmental genes in this case because some of the genes are not, so one of the genes are not available for this group of species, and the others, when tested with codon models, sometimes they fail to converge, and therefore I cannot get useful data out of it. But overall, with this data, we can conclude that the occurrence of accelerated evolution is associated with events. And so here's the discussion. So based on the current data, which of course is preliminary and limited, but beside what we can conclude is snake photoreceptor development genes analyzed in this study are indicated to have experienced accelerated evolution similar to what have been found in the snake phototransduction genes. And also the branches and clades where photoreceptor transmutation occurred are associated with the positive selection and elevated selection pressure. And in terms of the future directions, we can try some new codon models such as the phenotype-genotype model. And so this model can kind of link the phenotype, in this case would be the retinal composition, with the genotype, in this case would be the developmental genes. So we could further confirm this association, and we can also try some expand our data set to cone photoreceptor developmental genes and see if observe if they also experience similar selective shift. And so that's the end of my presentation, and also thanks for my lab, for the support of my lab members and supervisor. And so that's it.
Great, very cool talk. We don't have time for questions, unfortunately, but people can ask questions in the chat. You can ask them there if you wish.
Yeah, sure.
Hey, Laura, do you want to start up your talk?
Okay. Hi, everyone. I'm Laura Alencar. I am an associate research scientist at the Department of Ecology and Evolutionary Biology at Yale. And today I'll be presenting the first result of a collaborative project between the Munoz lab at Yale and Ueda lab at the Virginia Tech. So first of all, I would like to thank all my co-authors. So I'd like to start by showing you some beautiful images of fishes. Here we can see in this phylogeny of coral reef fishes that some lineages accumulate diversity way more rapidly than others, which here are illustrated by the different color branches color in the branches, where warmer colors represent.
We don't see the tree.
Sorry, right now we just see your title slide.
Oh, really? Okay.
So if you can make sure to just reshare and make sure that it clicks forward.
Let me try again. Sorry about that. It's not changing. Not changing. Okay, I'll do PDF. Sorry about that. I'll try to accelerate here. Okay. Okay, is it changing now?
Yep.
Okay, so those are my co-authors. This is the phylogeny of coral reef fishes, where warmer branches represent higher diversification rates. And here we have a similar example with squamates or lizard and snakes. In this case, the higher diversification rates are illustrated by the warmer colors in the circles. So these two examples represent actually a prevalent pattern in biodiversity. That is, evolution proceeds unevenly across the tree of life. And one of the major sources contributing for this diversity disparity is the access to different ecological opportunities that these lineages will face during their radiation, right? So events such as the emergence of a key innovation, like the evolution of viviparity or the toolpads, the invasion of new environments, or the extinction of antagonists such as competitors and predators, can create such opportunities and catalyze diversification, because lineages will take advantage of these vacant niches and they are able to generate more and more and more species. However, it would be great if life happened in such a simple, direct, and linear way, right? Where at least would be easier to quantify the cause and effect that these different ecological opportunities have in shaping the tree of life. But life is way more complex than that. And what we see instead is a complex network of processes shaping these diversification patterns. So lineages will encounter not one or two, but several sources of ecological opportunities during their radiation, as illustrated here in this hypothetical scenario. And so isolating the effect of these different ecological opportunities in shaping the diversification dynamics becomes very challenging. But we can do that, and this is actually our main goal in this study, where we wanted to untangle this complex network of process shaping macroevolutionary dynamics. So our first, what we wanted to do first is to isolate the different sources of ecological opportunities contributing to the diversification dynamics, explore the potential interaction between these sources, and by doing this, to come up with more plausible causal scenarios underlying the macroevolutionary dynamics. And specifically in this study, to do that, we focus on the Pleurodonta lizards, which comprise a group of mostly lizards from the Western Hemisphere, a little bit more than 1,000 species. It's a group that comprises the famous anoles, the horned lizards, and iguanas, and also the predominantly Andean genus, the Liolimus, which is actually going to be very important in this talk. So the first step we took to untangle this complex network of processes shaping the macroevolutionary dynamics of Pleurodonta was what we call the data-driven or the phylogenetic natural history step, where basically what we wanted to do was to first identify those lineages in Pleurodonta that have unique diversification rates, or in other words, those lineages that kind of stand out, that have super high rates compared to the background estimates. And by doing that, we found several instances where the diversification rates increased during the radiation of Pleurodonta. So here we have a phylogeny for the Pleurodonta, where warmer colors represent, again, higher rates. And we can see the red circles denote those instances where these rates significantly increase. And when we look more closely to those lineages with high diversification rates, we readily know that many of them share several life history, ecological, and geographical aspects. Many of them were viviparous, for example, were arboreal. Some of them occur in islands, have large body sizes. Many of them actually occur in mountains, and specifically I think it's very interesting, many of those mountains were specifically associated with the Andean mountains in South America. And these ecological opportunities or potential source of ecological opportunities were not randomly selected. They are actually widely discussed in the literature as being potential source of ecological opportunities, contributing to or shaping the diversification dynamics of not only lizards, but several other organisms. So the next step was actually to explicitly test the relationship between those potential source of ecological opportunities and the diversification rates. And we did several phylogenetic correlations to do that. And this analysis suggested that from these traits that we're interested in, two of them, that is reproductive mode and Andean mountains, had a relationship, a significant relationship with the diversification rates. Specifically, we found that vivipar species and or those present in the Andean mountains have higher diversification rates. But we knew that in Pleurodonta, many of those vivipar species also occur in the Andean mountains. So we're kind of like, are these two aspects truly independent source of ecological opportunities, independently contributing to this rate heterogeneity across the clade? Or maybe are they confounding each other in doing that? To tease that apart, then we use a combination of SSE models or the state-dependent speciation extinction models. So the first step we did, we use MISI, which basically is a method that quantifies the position, the number and location of different like rate categories that best characterize the rate heterogeneity across a given group. Specifically for Pleurodonta, MISI suggested that the 4 rate categories best explain the rate heterogeneity, here illustrated, represented by the different colors. Just to go very quickly to show you who belongs to each of these categories. So the first one comprises all the species of the Liolimus, those predominantly Andean genus that I mentioned in the beginning of the talk. The other one comprises Phymaturus, which is also predominantly Andean, and together with Liolimus and a few more species comprise the family Liolemid. The third category combines a Scelopodus subclade and the iguanas. And finally, the fourth category comprises the remaining lineages and will comprise what we call the background category. So the next step, we wanted to explicitly compare the rates between the lineages that occur, that are present and absent in the Andean mountains, and also between those lineages that are ovipars and vivipars within each of those rate categories. So just to make this plot a little bit clear, each pair of box plots represent one of these rate comparisons within each of these rate categories. So in the first analysis, we compare presence and absence in the Andean mountains, suggests that there is a positive effect of being in the Andean mountains in Liolemus and in Phymaturus. However, when we did the same thing, but now comparing ovipar species, the analysis suggests that only there is a slight increase in the rate associated with viviparity only in Liolemus. In case you're wondering that, Laura, but I can see that viviparity is also affecting Phymaturus over there. Just to make this clear, all Phymaturus species are vivipar, so we can't actually make this comparison. What you're seeing here is just an effect of the prior in there, so it's not empirical estimates. So to further then tease apart the role of reproductive mode and presence in the Andean mountains in shaping the diversification dynamics in Pleurodonta, we now wanted to compare these rates, the rate associated with these two traits, but now within the same analytical framework. And to do that, we chose to use the best clades to do that. And why I'm saying like, which is actually the Liolimus, the famous Liolimus. And why do I say that this is the best group to do that? So first, as Missy suggests, the first analysis, first SSE analysis that we use, all species of Liolemus belong to the same rate category. So this kind of helps us to control for potential hidden states in controlling those rates. Also, Liolemus is actually the only clade in Pleurodonta that has either vivipars and ovipar species, and also species that are present and absent in Andean mountains. So it's actually the only clade that we are able to perform this analysis. And on top of everything, it's also like one of the clades with the higher diversification rates, would make them even more interesting. So this analysis, the only significant result coming out from this analysis suggested that among Liolemus, those vivipar species have that are present in the Andean mountains have higher rates than those vivipar species that are not in the Andean mountains. So this led us to conclude that viviparity seems to have a minor effect, and it is actually the occurrence in the Andean mountain, the major contributor to the rate heterogeneity in the group. So this first set of results led us to conclude that some lineage-wide signatures are actually lost when we analyze subgroups like the Lyolimus. But what about the opposite scenario? What are the odds that we're actually missing signatures of traits that are potentially driving diversification rates because we're focusing on broad phylogenetic scales? So this question came to our mind specifically when we were thinking about body size, because body size is a relative metric, right? Being large or small is relative to the phylogenetic, ecological, or geographic context that a lineage belongs, right? So imagine that a crown giant, arboreal crown giant anole, is not that giant compared to another arboreal species like the iguana. However, the arboreal iguana is way larger than the viviparous terrestrial Phymaturus. However, the viviparous terrestrial Phymaturus is way larger than another viviparous terrestrial and very closely related species, the Lyolaemus pictus. So remember that in the beginning of the talk, I showed that we didn't find any relationship between body size and the rates when we analyzed the whole Pleurodonta together. However, when we analyze the species within each source of ecological opportunity separately, and also by combining different taxonomic scales, we do find that larger species, which here are represented by the brown triangles, tend to be associated with higher species diversification rates. So the second set of results suggests that some signals will fade at broader phylogenetic scales, but emerge at shallower ones. So this study kind of brings both organismal-focused conclusions and also macro-oriented conclusions. So first of all, viviparity contributes to diversification, but it's clear that the Andean mountains is inflating its impact. So we should think about viviparity as whole to be likely context-dependent, at least in squamates. Andean colonization drives Pleurodonta diversity, but also Lyolimus, and I will also add Phymaturus here, are clays that have their own unique dynamics. So there is something else about these two groups. There is, like, I would imagine something not related only to being viviparous and Andean that contributes to them to have like such huge or higher diversification rates. And finally, body size alone does not boost diversification in Pleurodonta, except when in synergy with other sources of ecological opportunities. And finally, universal signatures of ecological opportunities are elusive, so many will weakly affect diversification, while others promote diversification under way more limited conditions. So we should consider such context dependency when untangling the process generating the biodiversity patterns at large scales. I couldn't go over too much detail here, but we just published this study a few weeks ago. So if you're interested, feel free to download this open access, and I would love to have some feedback. I also went through over a lot of methods here, but we put this in a step-by-step fashion. So hopefully people will be able to use and apply the framework in their own study systems. I would like to thank everyone that showed up, all my co-authors again, people that helped in this project along the way, and our funding sources. And I will be happy to take any questions if I have time. I have no idea.
We have 1 minute left before the next talk, but I think the next speaker is not here.
Oh.
So we're going to ask questions. Omar, go.
Yes, hello. Congratulations, Laura. Great talk. And I have also already seen your paper, your nice paper. I have been working a lot with Stenocercus. You probably know also that taxon. And I'm always comparing Stenocercus with Lyolimus because they are very similar in morphology. They are both Andean, but they are quite different in biology and ecology and diversification. And I just have one question. Do you think another factor that will contribute to higher diversification rates is simply the presence of, or I mean, the absence of more community members? I mean, for example, Stenocercus lives with many other species of lizards, like genophthalmids and basically genophthalmids. And I don't know if, I'm pretty sure Lyolimus does not occur in sympatry with many other lizard groups. What do you think about this?
That could be potentially very good explanation, like to look at the communities and see like if they have many competitors, they're coexisting with less species. I think there are plausible explanation. I think the first thing that came to my mind when like knowing that like Liolimus and also Firmaturus have such high diversification rates, and like other Andean species or lineages doesn't show the same, like even close, right? What came to my mind, the community thing, it's like very interesting. I didn't think about it. What came to my mind immediately was the timing of arrival in the Andes. I don't have external circles like the age that they might have arrived, but I know that Liolaemus or Liolemids, I think it was Liolaemus, was already there. They were already there like way before the uplift started to like go faster. So my take on that was that they are just in the right place at the right time. But the community hypothesis is a good one for sure.
Great, thanks.
Thank you.
Thank you. Just checking that the next speaker is not here, correct? Like it could be under a different name or something.
It's Dylan, right? The next
Yeah.
one.
All right, in that case, we can take a break until the next talk at 6:45 conference time. If I was want to ask the previous speakers questions, see them still in the room, so you can ask away and chat or on voice.
Thank you.
Can folks see the cancellation slide okay?
Yep.
So Laura, I have a question. So you looked at net diversification rate. Do you have a sense of like the underlying, is it like a change in speciation rate or change in extinction rate or something else?
Yeah, so I mean, we actually did both, right? Not we did the diversification rates, we did the speciation rates, the extinction rates, the net diversification rates, but we're focusing more on the speciation rates because I mean that's what we could like actually get more real. I could rely. I mean, the hypothesis related to ecological opportunities, they are all based on speciation rates. So we were thinking more about the speciation rates anyway, and but yeah, we combined both. We did the SSC I presented here, if I'm not wrong, the net diversification results, but we have the speciation tip rates were all reflect more the speciation rate. So I would say that this is all based on the speciation rate side of it. I think if we're to think about the extinction, then I would think I would reframe some of those hypotheses because they were all relying on like being able to generate more species, I would say. But I mean, I would be interested to like think about the Firmaturus, for example, because it's such a, I don't know if you noticed the huge branch that it has. There's clearly a lot of extinction that like happened a while ago in there. So it clearly it was important for the group too. But yeah, the paper is all about speciation, I would say.
Very cool. I haven't read the paper yet, but I'm looking forward to it. Yeah, Hang, one question I had on your talk was some of your DNDSs were as high as like 11, which is huge. Where else does that happen in nature?
Well, I think that DNDS is actually, so 11 is pretty high, but it is not that high, according to my experience. So it's not for the snake photoreceptor genes, but I know for some fishes, like for their rhodopsin genes, they actually got like way more higher DNDS ratio, probably like
Oh, that's so
30,
cool.
40, yeah, it's way more higher. Because actually for the photoreceptor genes, for the developmental genes, they are relatively conserved given their important function. And especially these genes are also involved in other like neuronal structure development. So they're actually relatively conserved. But for some phototransduction genes such as rhodopsin, they well, I mean, they are like directly related to the like light sensing functions. So when there's a change in the visual ecology, there's actually a much greater magnitude of DNDS ratio
That's so cool. I had no
change.
idea.
So
That's neat.
yeah.
Were any of the snakes you looked at exclusively fossorial still, or again, were they all like ones that hunt visually?
For the snake I've been working on, I think they are all on visually, or at least they are terrestrial species. So I don't have.
Okay, yeah.
I think there are a few newly published genome for blind snakes. So those are fossorial snakes, but I haven't started working on them to like pull sequence out. So currently they are all terrestrial species, all aquatic species. But anyway, none of them are fossorial.
Okay, neat. Yeah, I didn't even think about the aquatic ones. Yeah, like we look at like sea snakes and see what they do. Or they have a different visual environment.
Yes, I kind of have kind of take a look at those aquatic species. I think they are showing some kind of elevation in DNDS ratio for something like only for like one gene something. So I'm not quite sure if that is like random event or if it actually means something. But yeah, that would be something interesting to take a look at.
Yeah. Great. Anyone else have any questions for the previous speakers? And I can't see hands up at the moment because I'm sharing my screen. So if you can just unmute yourself and speak, you wish. What cool things have you seen at the meeting so far? Anyone?
I'm afraid I'm on the other side of the world and I couldn't justify being up at 2 a.m. each night. So I haven't seen very much, to be quite honest.
Yeah, that makes sense. Yeah, I mean virtual. I mean, it's one of those things that like the in-person meetings tend to draw from mostly North America, just because geographically it's cheaper and such. But this meeting is like 3/4 of the people from outside North America, which is great, but it makes it
Oh,
so
it's amazing that you do it.
time
I'm so
zone.
appreciative of it.
Yeah.
I'm so appreciative of it.
It makes time zones hard. Yeah.
It would be fine if I didn't have other things to do during the day.
Yeah.
I was actually really pleased to find out that this was at this time. This is a pretty convenient time. It's only 8:40 in the morning here, so I could I could manage.
Nice. And all the talks are recorded. Most people have
Yeah,
opted out.
that's the other thing. They were recorded last year as well, so I caught up on quite a few things after the fact.
Yeah, I'm hoping to catch up with the talks, the recorded talks also, because I'm in another meeting at the same time. So I like they're having the reception just now. So it overlapped a bit the two meetings, but I'm glad that it will be recorded.
I'm glad you turned down food to give a talk. It was a good talk.
Oh, thanks.
I think one thing that to me was really cool was with the option to give also, like, some people were presenting recorded talks already. And that meant that they like got all of their team members to speak. So in a 10-15 minute talk, you had 6 or 7 people involved in a project that were speaking, which is rare. Usually you just hear the one person who's like chosen to spearhead things. And I thought that was pretty cool.
That's neat. Yeah. I don't even think that's allowed at the in-person meeting. I can't remember ever seeing a like a tag team talk.
I haven't seen it for evolution, but I know one of the coolest talks I've listened to was actually someone who was like a postdoc, and they just stepped off the stage and let their intern kind of talk about the cool results that they had done on their independent project, which was really fun.
That's really good. Yeah. Especially at conferences where you don't put microphones on people, it's very feasible to do it. Yeah. Let me stop sharing.
Where are you at the moment, Brian?
I'm in Oak Ridge, Tennessee, in the US.
Cool.
Yeah, I went
Nice
lucky.
backyard.
Thanks. Yeah, we're lucky. Like, well, fireflies come through. We had a bobcat come through. It's like really cool biodiversity, even though being pretty much suburbia.
Yep.
But since I'm uh doing the not the mentoring thing though, the hosting thing, I can no longer be in my hammock because it's too informal. So I like sit at a table. All right, so we'll start in 20 seconds with Maya's talk, which is recorded. And Laura, you're launching the talk.
Yes, 1 second. Zoom's asking for a password to share audio. Okay, I'm gonna try it like this. Otherwise, I need to download Zoom audio, something different. So we're gonna see if the volume is not okay. Let me know, and then I can troubleshoot it.
Not hearing anything yet.
Here, let me try this again. Ooh,
Hello, my name is Maya Pelka.
Yep,
I am
good.
a graduate student at the University of British Columbia, Canada. I'm sorry I can't be there today to give you this talk live. I'm currently in Japan with one of my co-authors, Kevin Wakeman. So I figured it would be better to pre-record this talk as opposed to trying to give it and risk technical difficulties. So today I'm going to tell you a little bit about a really cool and charismatic protist or microbial eukaryote that possesses epibiotic bacteria and mitochondrial-related organelles. But before I tell you about Pleiosaurus cubiculata, I think we should introduce bacterial symbioses as a whole. Bacterial symbioses are incredibly widespread in microbial eukaryotes. On the side here, you can see a tree of eukaryotes, which includes all of the major groups, including plants, animals, fungi, and all of the major groups of protists. You can see that in almost every single major protist group, there are representative taxa that possess some form of bacterial symbiosis. And these bacterial symbioses are diverse in their taxonomy, they're diverse in their role, whether or not they are beneficial, commensal, or harmful, as well as in their localization, where we can have bacteria that are present on the external surface of a cell. These are known as epibiotic bacteria. We can have bacteria that are present within the cytoplasm of a cell. These are considered endobiotic or endosymbiotic bacteria. And we can also have endonuclear bacteria that are present within the nucleus of the host cell. My major group of interest is within the supergroup Discoba. Discoba is well known for its pathogenic organisms, such as Naegleria fowleri, also known as the brain-eating amoeba, which I'm sure many of you have heard of, as well as the kinetoplastids, which include the trypanosomatids, which are also pathogenic to humans as well as to many farm animals and livestock. Within Discoba, there is another major group known as the Glenozoa. The Glenozoa consist of 4 groups, including the symbiontids, which are characterized by a coat of epibiotic bacteria and mitochondrion-derived organelles, and the symbiontids typically thrive in low oxygen marine sediment. The kinetoplastids, which are both parasitic and free-living organisms and include those pathogenic organisms I just mentioned. The diplonemids, which are the most abundant cells in the pelagic ocean environment, and of course the euglenids, which is my major group of interest. Euglenids are incredibly morphologically and functionally diverse, with both heterotrophic and phototrophic lineages. The heterotrophic lineages consist of both phagotrophic and osmotrophic genera, and the phototrophic lineages are capable of photosynthesis through the acquisition of a secondary algoplastid. The phototrophic lineages are really well understood because it's very easy to culture these organisms. However, the heterotrophic lineages, and specifically the phagotrophic lineages, are very poorly understood and very poorly characterized, despite being really morphologically interesting and diverse. Within the euglenids, there are only 2 species that possess epibiotic bacteria. And just to recap, epibionts are bacteria that are present on the external surface of the cell. And even though we do know that these 2 species exist, we know very little about them. In fact, for one species, Leposynclus helicoideus, we only have one SEM, and that's it. We have no molecular data, no TEM of these bacteria. We don't really know anything about the system. For Delactosoma, all we have is this really, really terrible light micrograph. So we really do not know a lot about epibiont possessing euglenids, despite the fact that we do know that they exist. My species of interest, Pleochus scribiculata, was described in 1990 in Australia by Larson and Patterson. And since then, it has been completely untouched by the literature with no molecular or ultrastructural data. However, I have found this cell to be relatively abundant in the benthic environment in British Columbia at multiple sampling sites, and I have been able to collect quite a few of these specimens for electron microscopy. One thing that's really interesting is that these cells are characterized by this roughened cell surface, which is actually the name of this organism, scribiculata, actually refers to this roughened cell surface because scribiculata means sandpaper, which is really interesting. Now, in scanning electron micrographs, where you can observe the external surface of tissue or a specimen, we can see that scanning electron micrographs of these cells, of Pleochus scribiculata, demonstrate a coat of epibiotic bacteria that completely covers the cell surface of these cells, which is really, really cool. Bacterial symbiosis to this extent, where the cell surface is completely covered by epibiotic bacteria, has never been described using electron microscopy in the euglenids. So this is really, really exciting. Transmission electron micrographs, which show the internal structures of cells and shows the structure of organelles, demonstrate that these epibiotic bacteria are completely covering the surface of the cell and that they are associated with the cell surface through a glycocalyx or a mucilaginous layer. Here, I would like to emphasize that this is all single cell electron microscopy. So these cells are not in culture. They are found in semi-anaerobic sediment, where we have to go out into the field, collect sediment, and then specifically isolate these cells and process them all the way through electron microscopy, which includes the embedding stages, fixation, embedding, microtomy, and imaging. So this cell actually in the corner of this image is the exact same cell we are looking at in these electron micrographs. Not only do these cells possess epibiotic bacteria, but they also possess mitochondrion-related organelles, or MROs. Mitochondrion-related organelles are derived mitochondria that are typically very reduced in function and don't function the same way that a normal mitochondria would. These mitochondrion-derived organelles typically lack cristae. And so when I was observing this cell using transmission electron microscopy, I was unable to find any mitochondria that were typical in ultrastructure with many cristae. These mitochondrion-related organelles were also found near the periphery of the cell, which is particularly interesting because it has also been observed in other euglenid species, particularly Euglenia gracilis, which is a model organism that when cultured under anaerobic conditions, the mitochondria are typically situated near the cell surface. Whereas when these cells are cultured under aerobic conditions, the mitochondria are situated relatively uniformly distributed throughout the cell cytoplasm. Within Euglenozoa, there are other cases of these mitochondrion-related organelles that are situated near the surface of the cell and potentially associated with bacterial symbionts. So I mentioned a group known as the symbiontids earlier. And these symbiontids are characterized by this coat of epibiotic bacteria and mitochondrion-related organelles. However, these symbiontids are phylogenetically independent of Pleochus scribiculata. So though they are part of the same major group, the Euglenozoa, these are not closely related, and the mitochondrion-related organelles in Pleochus scribiculata did arise independently. What's really interesting about the symbiontids is that they're found in the same low oxygen sediment as Pleochus scribiculata. And they possess these mitochondrion-derived organelles that are situated right underneath the cell surface and are associated closely with the bacterial symbionts present on the cell surface. You can see here is another example of a symbiontid. This is Hospitis baccati, which was described by our lab. And you can see here that the mitochondrion-related organelles are situated right underneath the cell surface where there are rod-shaped epibionts. It has been hypothesized in the system that there may be some metabolic exchange between the bacterial symbionts and the mitochondrion-related organelles, which may allow these cells to thrive in this low oxygen environment. So it would be interesting to determine whether or not this is a similar relationship, or this relationship is occurring in Pleocia scribiculata based on the localization of the mitochondria. So I know that I mentioned that there was no molecular data for Pleocia scribiculata so far. So that really brings up the question, well, could it potentially not be a pleocyte or not even a euglenic? Maybe it's symbiontic, considering that it has these epibionts and it has these mitochondrion-related organelles. However, the ultrastructure of Pleocia scribiculata is incredibly consistent with that of typical pleocytes. To start off, Pleocia scribiculata possesses these proteinaceous pellicle strips, which are thick strips that have underlying microtubules which are capable of sliding along each other and allowing some euglenids, depending on the particular group of euglenid, to have this metabolic flexible motion, which allows them to consume larger prey. These pellicle strips are considered a synapomorphy for euglenids, which means that no other group within the euglenozoa should have them. They are really, really specific to euglenids. And you can see here in this SEM of Pleocia costata, which is another pleocid, that there are these clear and distinct pellicle strips. And in fact, Pleocia scribiculata, my group or my species of interest, has 10 pellicle strips, which is consistent for most pleocids. Pleocia scribiculata also has the classic pleocid feeding apparatus with 2 rods, and see right here, and 4 veins that wrap around the prey as the cell is ingesting its prey. And this is known as a type 2 feeding apparatus, which is consistently described in pleocids and not really described at all in symbiontids. Last but not least, the flagella and flagella root system of Pleocia scribiculata is consistent of that with pleocids. Typically, when you see pleocids, they will have a much, much thicker ventral flagellum and a much thinner dorsal flagellum because they glide along their ventral flagellum, and the dorsal flagellum doesn't do very much. This is very clear in this TEM right here, where Pleocia scribiculata had this really, really thick, dense ventral flagellum and a really, really thin dorsal flagellum. There were also flagellar hairs present, or not always described in symbiontids, but always are very consistent in pleocids. So the next step to this project is to determine the identity of these bacterial symbionts, and that involves a few different steps. First, we need to barcode these bacterial symbionts to determine their molecular identity. But barcoding can typically barcoding results can be affected by contamination. So it's important to also confirm the identity of these symbionts using something called fluorescence in situ hybridization. Fluorescence in situ hybridization allows you to tag a fluorophore to a particular region, RNA or DNA region of interest, to confirm the identity of whatever it is you're looking for. So I have had successful hybridization of a universal bacterial probe to the epibionts of Pleocia scribiculata, which is sort of the first step to getting successful hybridization of a taxon-specific fish probe. 16S rDNA barcoding results do indicate that these epibionts are gamma proteobacteria. So if this is in fact the case, when I do taxon-specific fish with a gamma proteobacteria probe, we should see similar patterning and signal with that probe as we do with both the universal bacterial probe and with DAPI stain. So the main takeaway from this talk is that Pleocia scribiculata possesses independently derived bacterial symbionts and mitochondrion-related organelles. I know that I talked a lot about bacterial symbionts being widespread across the eukaryotic tree of life, but mitochondrion-related organelles are not. This is rare, and the mitochondrion-related organelles are rare, and they do not typically occur independently frequently across the tree of microbial eukaryotes. So it's really interesting that there are mitochondrion-related organelles that have been acquired independently twice within the euglenozoa, if not more times. So this brings up the question, well, is there some form of metabolic integration between the bacterial symbionts and the host cell? In order to answer this question, we need to perform 18S rDNA barcoding or transcriptomics of Pleochus cubiculata to confirm the molecular identity. Fluorescence in situ hybridization experiments with a specific gamma proteobacterial probe will confirm the identity of the bacterial symbionts, and eventually genomics of these epibionts and the mitochondria of Pleochus cubiculata will be able to reveal and unravel the complex functional relationship between these epibionts and these mitochondria, whether or not there might be some sort of metabolic integration. Last but not least, I'd like to just thank all of the members of my lab, including my supervisor, for all of his help, and our co-author at Hokkaido University, Dr. Kevin Wakeman. Since I'm not able to be there to answer any questions, please feel free to email me at this email address if you have any further questions or thoughts about my research. Thank you.
Great. And let's let Adam start so we don't lose too much time.
Hello, my name is Maya Palka. I am a graduate student at the university.
Great. Go for it. You're still muted.
There we go. Can you hear me now?
Yep.
That's great. Thank you. All right. My name's Adam Bowden. I'm a PhD student in biological anthropology at the Australian National University. And I'm going to talk to you about a paper I've written with my supervisor, Phil Kavanagh, from the University of Canberra and the University of South Australia, titled Sex Differences in Romantic Love, an Evolutionary Perspective. So what is romantic love? Bowden-Cushnick defined romantic love as a motivational state typically associated with the desire for long-term mating with a particular individual. It occurs across the lifespan and is associated with distinctive cognitive, emotional, behavioral, social, genetic, neural, and endocrine activity in both sexes. Throughout much of the life course, it serves mate choice, courtship, sex, and pair bonding functions, and it can best be described as a suite of adaptations and byproducts that arose sometime during the recent evolutionary history of humans. It's essentially that thing that happens at the beginning of a romantic relationship where you go a little bit crazy and think your partner is the best thing ever. It can happen in the absence of a romantic relationship and rarely, sometimes after many years or decades. It's sometimes referred to as being in love by the general population or as passionate love by some psychologists. So what are psychological sex differences? Those are differences between females and males that basically emerge when the sexes face different adaptive challenges. Sexual selection is the evolutionary process through which psychological sex differences evolve, and they're common in reproductive traits, particularly mate preferences, sociosexuality, which is a desire for short-term mating, and sexual regrets. We don't know very much about sex differences in romantic love, although there have been a few small studies. And from these aren't all of them, but these are indicative of the findings. And from them, we know that males tend to fall in love slightly more often than do females. We know that males tend to say, I love you first in a romantic relationship. And there's some indication from some studies not designed to measure this, but that did measure this, that females experience romantic love slightly more intensely than do males. So what did we do in the current study? Well, we noted that no study has ever investigated sex differences in romantic love in a sample exclusively experiencing romantic love. So the existing studies had used samples of people in romantic relationships, some of whom were probably in love. So we investigated the univariate and multivariate sex differences in a sample of people experiencing romantic love. And I'll just note this paper is currently under revision at the Biology of Sex Differences. We got our data from the Romantic Love Survey 2022, which is a sample of 1,556 English-speaking young adults aged 18 to 25 from 32 OECD countries and South Africa. It was collected online using Prolific between October and December 2022, and it is the world's largest data set of people experiencing romantic love. It covered a range of topics, and it's now a longitudinal study with data collected every 6 months for 2 years. The metadata and the data is posted on the Love Consortium Dataverse. And this heat map just shows where we got our participants from, mainly, as you can see, from Europe, North America, and South Africa. We did oversample South Africa, Poland, and Mexico to try to make our sample a little less weird. That is Western, educated, industrialized, rich, and democratic. So the first thing we did was look at univariate sex differences, and we looked at number of times in love, love progression, which is a measure of when individuals fell in love relative to when they started their romantic relationship, the intensity of romantic love, obsessive thinking, and commitment. And what we've got here are the means and standard deviations, as well as some test statistics. And on the right, the most important statistic that we've got is Cohen's d, which is an indication of effect size. So as you can see, men had fallen in love slightly more often than had females. Men tended to fall in love 1 month earlier than did females. Women experienced romantic love slightly more intensely than did males. Women experienced obsessive thinking about their partner notably more than did males, and women were ever so slightly more committed to their partner than did males. But as you can see from the effect size in Cohen's d, all these effects are quite small, and really only obsessive thinking is anything of meaningful magnitude. So after that, what we did was run a series of hierarchical linear regressions where biological sex was the predictor variable, and we controlled for a number of suspected covariates. And as you can see, when we did that, the effect of sex reduced or disappeared entirely. So biological sex explained 1% of the variance in number of times in love. When an individual fell in love relative to when they started their relationship, sex explained 0.5% of the variance in that. Sex explained none of the variance in the intensity of romantic love. Sex explained 2.1% of the variance in obsessive thinking, and sex explained none of the variance in commitment. So quite notably, intensity of romantic love and commitment don't seem to be influenced by biological sex. So the main findings are that small univariate sex differences from most variables were found, but the factors other than biological sex explain these differences more than does biological sex. We know that the social environment interacts with biology to cause these sex differences, and this was particularly in the case in some variables where an association was found with gender inequality, specifically number of times in love and obsessive thinking. We found that the more unequal a country was, the more likely females were to score higher on number of times in love and obsessive thinking. So what are the implications for the evolutionary history of romantic love? Well, very small sex difference indicate that the primary adaptive challenges may have been largely the same for females and males. And we suspect that is that romantic love serves to promote the survival of the fetus and infant via survival of the mother. There's also some meaningful differences that emerged that were different, indicated different adaptive challenges. So males fall in love more often, and males fall in love 1 month earlier. And I'll explain that a little bit when I talk about the functions. So surprisingly, there's also a bit of implication for the mechanisms of romantic love. We know that this very small sex differences indicate that genes that regulate the onset and expression of romantic love are probably on non-sex determining chromosomes. And there is some indication that the mechanisms that are fine-tuned for mother-infant bonding in females may explain some aspects of sex differences. For example, obsessive thinking. There is a theory that romantic love evolved by co-opting mother-infant bonding, and that would mean that females are more attuned to performing some of the expressions that romantic love entails, particularly obsessive thinking. There's also a small implication for the functions. Romantic love serves as an honest signal of commitment for males more often than for females, and there is some indication that romantic love plays a role in proactive courtship more often than males. More often in males than for females, and this is because they fall in love 1 month earlier and can undertake that sort of stuff. So generally, I thought this was an interesting study. Got some references there, short and sweet. If you've got any questions, be happy to answer them.
And we have time for questions. So anyone have questions for Adam?
Does it sound like it? Very good.
All right, thanks. We'll hold before the next talk, so people who are switching between sessions can not show up late inadvertently.
I'm seeing one question in the Q&A. That's
Oh,
asking, how do you account for diversity in biological sex, such as hormones, chromosomes, etc.?
Yeah, good question. I'm not really well versed in evolutionary theory. Our data basically in the survey, we just asked what individuals' biological sex was, and it was that simple, and they could choose between male, female, or intersex, so that's how we did it. In terms of what might be causing these differences. Could be the mechanisms that are associated with a range of factors that tend to be different between females and males.
I have maybe one quick question for you, if we have time. I think we have time, right, for Adam?
yeah, you can.
I think it's a really cool study. Can you hear me all right first? I don't know if that's...
Yes, I can.
Or I've been filling with those. I was wondering if you found anything, or if your analysis could actually find differences among the sexes in the variability around the estimates that you report. So you might not have any mean differences among the sexes in the variables that you saw, but maybe males are all over the place and females are a lot closer to the average. Do you have any of these signatures or types of results maybe in your...
Sure. So we could definitely have a look at the data and do some post hoc analyses. The standard deviations were reported for the univariate sex differences, and the standard deviations looked relatively similar. I mean,
Okay.
that's just one indication, but we could probably eyeball the data and see if we can find any other relationships. And maybe I'll spend an afternoon one day doing that, because that would be a nice little addition to the discussion section if we could find something interesting. Thank you for that.
Definitely. Great talk.
There's another question. What evolutionary hypotheses are you testing?
So it was an exploratory study because there'd never been a study investigating sex differences. So the theory that drove it is that psychological sex differences occur as a result of sexual selection in circumstances where females and males experience different adaptive challenges. We know very little about the evolutionary history. So what we sort of assumed was that if females and males faced different adaptive challenges, we would find large sex differences. We found small sex differences, which indicated that the adaptive challenges were probably very similar for both females and males throughout the evolutionary history of romantic love.
Another question, did the study only include heterosexual relationships?
No, it didn't. So our sample was quite young, and there were a lot of university students. So it was actually a very high proportion of bisexual people, especially bisexual females. I think something close to 25% of our sample identified as non-heterosexual. I think about 5% of males were homosexual, about 6 or 7% of females were homosexual, and a large proportion were bisexual.
All right, thank you. Let's set up for the next talk. You want to start sharing your screen?
Okay.